Covishield. The coronavirus is studded with proteins that it uses to enter human cells. These so-called spike proteins are used to create the Covishield vaccine. The researchers added the gene for the coronavirus spike protein to another virus named adenovirus. Adenoviruses are common viruses that typically cause colds or flu-like symptoms. Covishield for COVID-19 is more rugged than the mRNA vaccines from Pfizer and Moderna. This is because Covishield uses DNA. DNA is not as fragile as RNA, and the adenovirus's tough protein coat helps protect the genetic material inside. After the vaccine is injected into a person's arm, the adenoviruses bump into cells and latch onto their surface. The cell engulfs the virus and pulls it inside. Once inside, the adenovirus travels to the nucleus where the cell's DNA is stored, and the adenovirus pushes its DNA into the nucleus. Now the cell's molecules begin assembling spike proteins. Some of the spike proteins produced by the cell form spikes that migrate to its surface and stick out their tips. These protruding spikes and spike protein fragments can then be recognized by the immune system. When a vaccinated cell dies, the debris contains spike proteins and protein fragments that can then be taken up by a type of immune cell called antigen-presenting cell. When other cells called helper T cells detect these fragments, the helper T cells can raise the alarm and help other immune cells to fight the infection. Helper T cells, along with other immune cells called B cells, identifies coronavirus spikes and will start to proliferate and pour out antibodies that target the spike protein. The antibodies can latch onto coronavirus spikes, mark the virus for destruction, and prevent infection by blocking the spikes from attaching to other cells. The antigen-presenting cells can also activate another type of immune cell known as killer T cell to seek out and destroy any coronavirus-infected cells that display the spike protein fragments on their surfaces.